Assalamualaikum warahmatullahi wabarakatuh. Hi everyone. We meet again in biology lecture with me, Madam Hida. Our topic for this video is mutation. Previously, we have learned about 7.1 mutation classification and types. In this video, we will focus on 7.2 gene mutation. At the end of this video, students should be able to achieve the three learning objectives. Number 1, students should be able to define gene or point mutation. Number 2, students should be able to state the 4 types of gene mutation, which involve base substitution, base inversion, base insertion, and base deletion. Number 3, students should be able to state the effect of base substitution, base insertion, and base deletion. How is the change in DNA base sequence causes the change in the codon sequence of mRNA and amino acid sequence of protein? Recall back what we have learned about central dogma. Central dogma describes the ways in which genetic information is transferred from DNA into mRNA via transcription and from mRNA into polypeptide chain or protein via translation. To have an overview idea on how gene mutation affect the production of polypeptide chain or protein in a cell, let's recap on the process of transcription and translation in a eukaryotic cell. Specific bases sequence in DNA template strand will be transcribed into a single strand of pre-mRNA via transcription process in the nucleus. The pre-mRNA will undergo RNA processing and become mature mRNA. The mature mRNA will move from the nucleus to the cytoplasm. In cytoplasm, the mRNA will undergo translation and will be translated into polypeptide chain. According to Campbell, gene mutation is defined as the change in a single nucleotide pair in DNA base sequence. Meanwhile, Mader stated that a gene mutation is the altered gene where the mutated basis sequence of DNA differs from the original sequence. Gene mutation occurs spontaneously, which involves the mistake in base pairing during DNA replication of the cell. Changes in the DNA base sequence will lead to changes in the codon sequence of mRNA, thus changes the amino acid sequence and resulting in the production of different protein. There are 4 different types of gene mutation: base substitution, base inversion, base insertion, and base deletion. Base substitution is the replacement of one nucleotide and its partner with another pair of nucleotides. The example of disease resulted from base substitution is sickle cell anemia. For the normal red blood cell, the shape is spherical and flexible, while the sickle cell is sickle-shaped and rigid. Sickle cell anemia is the result of mutation in beta-globin gene of hemoglobin. Normally, the flexible and spherical red blood cells move easily through blood vessels. But in sickle cell anemia, the red blood cells are shaped like sickles or crescent moons. These rigid and sticky cells can get stuck in small blood vessels, which can slow or block blood flow and oxygen to parts of the body. For the normal red blood cell, thymine base pairs with adenine in DNA. During transcription, thymine base transcribes into adenine. Codon GAG will undergo translation and translated into glutamic acid in polypeptide chain of normal hemoglobin. Contrastly, in sickle cell anemia, thymine base has been replaced by adenine in DNA sequence. During transcription, adenine will be transcribed into uracil. Codon GUG will be translated into valine in sickle cell hemoglobin. To summarize the base substitution, it happens when the nucleotide pair of DNA replaced by another pair of nucleotide, resulting in the changes of codon in mRNA and synthesize different protein. However, some base substitutions have no effect on the encoded protein, that kind of mutation known as silent mutation, and the rest has an effect on the encoded protein, and they are namely as missense mutation and nonsense mutation. Silent mutation has no observable effect on the phenotype because it changes the nucleotide pair in DNA-based sequence and transforms one codon into another that is translated into the same amino acid. This happens due to the redundancy property of the genetic code, which means one amino acid can be coded by several codons. As we can see from the given diagram, guanine base in DNA template strand will be transcribed into cytosine base in mRNA. The codon GGC in mRNA will later be translated into glycine amino acid. When the base substitution occurs, adenine base replaces the guanine in DNA. Adenine will be transcribed into uracil base in mRNA. The codon GGU in mRNA will then be translated into the same amino acid, which is glycine. Such a change is an example of a silent mutation. Base substitution that change one amino acid to another one are called missense mutation. From the given diagram, cytosine base in DNA template strand will be transcribed into guanine base in mRNA. The codon GGC in mRNA will later be translated into glycine amino acid. When the base substitution occurs, thymine base replaces the cytosine in DNA. Thymine will be transcribed into adenine base in mRNA. The codon AGC in mRNA will then be translated into serine amino acid. Base substitution can also change a codon for an amino acid into a stop codon. This is called a nonsense mutation. Most nonsense mutations lead to non-functional proteins. To make it clear, let's take a look at the example. In the diagram, thymine base in DNA template strand will be transcribed into adenine base in mRNA. The codon AAG will be translated into lysine amino acid. When the base substitution occurs, adenine base replaces thymine in DNA. Adenine will be transcribed into uracil, producing UAG codon. UAG is one of the stop codons, and it causes translation to be terminated prematurely. The resulting polypeptide will be shorter than the polypeptide encoded by the normal gene. Base inversion is another type of gene mutation. It happens when two nucleotide pairs or more in the DNA are inverted. As any method, the nucleotide pair of cytosine guanine and thymine adenine is reversed end-to-end, producing the mutated DNA base sequence. Base insertion is the addition of nucleotide pairs in a gene. For example, the normal DNA base sequence is 3' A, C, T, A, G, T, C, G, G, 5', complementary with 5', T, G, A, T, C, A, G, C, C, 3'. When the new nucleotide pairs were inserted within the original reading frame, it will cause the reading frame shift its position from the point of insertion, producing the longer mutated DNA base sequence. Base deletion defined as the loss of nucleotide pairs in a gene. For example, the original DNA base sequence is 3' A, C, T, A, G, T, C, G, G, C, A, T, 5', complementary with 5', T, G, A, T, C, A, G, C, C, G, T, A, 3'. When two nucleotide pairs were deleted from the original reading frame, it will cause the reading frame shift its position from the point of deletion, producing the shorter mutated DNA base sequence. Insertion and deletion of nucleotides within the original DNA sequence may alter the reading frame of genetic base sequence in DNA. Such a mutation, we call it as frameshift mutation. It occurs whenever the number of nucleotides inserted into the original DNA base sequence or deleted from the original DNA base sequence is not in multiple of 3. Many of these insertions and deletions start in the middle of a codon, thus shifting the reading frame by 1 or 2 bases, resulting in all the nucleotides downstream from the point of insertion or deletion will be improperly grouped into codon. This diagram shows how the basis sequence of DNA template strand has been transcribed into mRNA codon and translated into a sequence of amino acid consists of methionine, lysine, phenylalanine, and glycine. What happens when the insertion or deletion occurs within the reading frame? Insertion or deletion of base not in multiple of 3 will shift the reading frame by 1 or 2 bases. This effect known as frameshift mutation. Frameshift mutation can be described by these two diagrams. In diagram 1, the insertion or deletion of base changes a codon for an amino acid into a stop codon. The polypeptide produced will be shorter than the polypeptide encoded by the normal gene. Whereas in diagram 2, it shows the insertion or deletion of base which also may change the codon sequence that code for different amino acid from the point of insertion or deletion. This will produce the new protein that may become non-functional. In diagram 3, it shows when the insertion or deletion is in multiple of 3. That will cause no frameshift. However, the insertion of triplet bases will lead to an extra amino acid encoded, whereas the deletion of triplet bases causing one amino acid is missing. To summarize the frameshift mutation, when insertion or deletion occurs, it will change the coding sequence of DNA, therefore lead to changes in codon and amino acid, resulting in different polypeptide chain or protein produced. The effect of frameshift mutation is usually harmful to human. Example of disease which caused by frameshift mutation is major thalassemia. That's all about subtopic 7.2 gene mutation. In the next video, you will learn about 7.3 chromosomal mutation. Till we meet again. Bye-bye.